Okay, thanks to several slides. To begin with, several slides are from Jan Ayofran, who's now professor at Bar-Ilan University and Boston University. Slides also come from Punta, who is now in Paris, and I have to update that. It's not true. So Avner Schlesinger is right, is a professor at Mount Sinai, but Punta is in London. Karl Svrzyński is in the Genome Center in New York. And some slides will come also from Jana Bromberg, who is at Rutgers University. Okay. In some sense, this introduction is a repetition from what happened to what some of you heard in last semester. A little bit I already brought it, but let's begin here. How does life begin? How do you imagine that life begins? So, you know, there's sort of the conditions are there that life can begin a billion years ago. But how does it start? What is sort of starting life? Any idea?
The basis of, for example, amino acids or the basis of DNA coming together to form a sequence? Actually,
you're very close, but more precisely, the assumption is that none of the two molecules that you're naming, it's neither amino acids nor nucleic acids. Well, it is actually nucleic acid, but the idea really is life begins with RNA. Because RNA, and that is here, for instance, formulated, Tom Check is one of those, Atkins. So the idea is that RNA, unlike DNA, has stability and can self-replicate more easily. So now RNA at some point begins this, and maybe there is a transition through DNA, but ultimately RNA needs some enzymatic activities, and there you're absolutely right. So it needs to bind actually amino acids. So it needs to sort of bind what you can consider protopeptides. So peptides is what we call short stretches of protein. Short stretches of protein, the idea really is this is what they used in order to do enzymatic function activities, to do chemical processes that they could not do otherwise on their own. So your idea is right, so there's nucleic acids and amino acids come together, but the idea is a little bit further. So there really is the idea there's a molecule that you could sort of call proto-life or whatever you call it, so the precursor to life, which really is RNA, which replicates itself, and it has sort of enslaves proteins in some sense, or it uses proteins to do some processes that the RNA world alone could not do. So it enriches it well. Now, if that were right, that proteins sort of do the job, and then ultimately what I told you already is today information is stored in the DNA. There is a transcript RNA. The RNA is functional in the cell, but most of the function, essentially the machinery of life, are the proteins. Now, if the proteins sort of were sort of the initial peptides that came were those that helped RNA do something, what would you expect of the proteins today?
What was the question again?
So if this image is right, again, it's an assumption, okay? RNA begins life. This is the one model that currently is believed. And if that model were true, and if RNA really need to enslave amino acids or protein or peptides to do some enzymatic activity. Now the proteins that we see today come from those, right? What do you expect in this set of proteins that we see today? Do you believe there is any evidence? And if so, what sort of thing would you expect to see? Any idea?
There must be something so that from proteins we can get back to RNA as well.
Yes, what do you mean by get back to?
I mean, like by transcription.
Oh, so turning a protein into RNA. That's an interesting idea. No, but actually think about it. So the RNA needs helper. The helper doesn't need to sort of turn back into an RNA, right? Mm-hmm. The RNA needs a helper.
So you mean the same product of RNA, which are protein, are those needed to RNA needs protein to make a protein.
Yeah.
So it's like
not only to make a protein, it needed proteins to do all kinds of things. So needing protein to make a protein, that comes later. In fact, if you look into a cell, the largest apparatus, so to speak, that is actually doing something that is relatively protein-free. There's half of it is proteins, but half of it is actually RNA, is the ribosome. That's where proteins are made. So proteins are still made today in every one of our cells, not without proteins, but with a lot of RNA. So that still is true. Now, so again, the slaving, or the initial idea of using proteins to do some chemistry is not necessarily the chemistry of doing proteins. All kinds of other things, all kinds of enzymatic activities. Now, but my question is, if that really were true, what would you expect of the proteins that we see today? Or what could you try to look for in the proteins that we see today? So essentially RNA finds amino acids, puts them together, makes simple proteins, peptides, and out of these evolve over 1 billion years the universe of proteins that we are going to discuss in this lecture. What do you believe you will see in this universe of protein structures if that initial idea that essentially RNA sort of moved around and found the part of peptides that could help it to do some chemistry? What would you expect of the proteins we see today? Well, so the idea is that you would expect something that reveals that once it bound RNA, that you would see something such as when you look at the protein structure from today, that you can sort of go back and see the core of this thing. At some point, for instance, did bind RNA or did something that was helpful to RNA. So we have 20 different amino acids. If that model were true, we would expect that there are many more amino acids available in nature. So why is it that only these 20 essentially are in our bodies? There are a few more, but they don't really count because it's not a lot of those. But essentially we use only 20 of, I don't know how many possible amino acids there are, but many more than 20. Why those 20? And the answer then would be because those 20 help exactly for the kind of chemistry RNA needed, and those 20 are enriched in, for instance, RNA binding. And if that is true, then you have an idea that in fact this contributes to this model. And one person, I don't know, I somehow lost the photo of Andrei Lupas. So Andrei Lupas made that, he's a director of the Max Planck Institute in Tübingen, and he essentially made that his life's work to go through particular footprints of proteins and computationally and experimentally prove that there is sort of a core that goes back to the RNA world days. So he essentially assembles a lot of evidence that in fact that's exactly what we see today. And the proteins always, it's never causal, right? You can never prove, but at least he aggregates a lot of evidence, and the story becomes stronger and stronger over the years. And those are remarkable stories about evolution of proteins. Now, we have something like an ocean. I mean, most likely we believe this begins in water. We have some RNA, and we have some proteins. What else do you need?
What's ocean?
Well, it's sort of the thing with the salty water where some of us swim once in a while. 70% of the Earth's surface is that thing. I'm sorry, that was unfair. It was highly unfair. But again, you know, I'm calling it ocean because ultimately life evolved in the ocean, right? But think about it as water, as just these things are pretty small, so a drop of water is enough to make them feel like in an ocean, right? A drop of salty water. A tear of you is enough to make this RNA feel that it's in an ocean because it's completely immersed by water. Anyway, my question is essentially, so you need, I could have written water, but ocean sounded better when I wrote this. Now that you asked the question, I should have written water. I'm sorry. So you have water, you have RNA, you have some proteins. What else do you need to sort of make life happen? Yeah, well, actually, no. You don't really need it. So today we cannot imagine. So we are DNA-based life forms, carbon. So we know essentially of. Now things get very complicated, but we essentially know of no life form without DNA. So in that sense, you're absolutely right. But So in this, let's call it then proto-life state, where we have RNA and proteins, there is no DNA at this point. So DNA is a mechanism to have an easy long-term storage of information. Because RNA is going to be eaten, it's not as stable as DNA. So if you put the information into DNA, you can put that somewhere, it holds very long. Again, very long does not mean on the scale of of planet Earth, of life. So DNA does not survive a billion years. So getting DNA out of fossils is highly non-trivial because it is sort of eaten up. Why is it eaten up? Because the air is full of bacteria, the air is full of enzymes that are free floating around and would just eat up nucleotides. Or have a chemical reaction. It depends a little bit on how you formulate this. But now, again, we have RNA, we have proteins. What else do we need to, yeah?
Probably we need something that separates the ocean from our organisms.
We need the idea of self. Yes, we need a separation. We need to put it somehow in a bag. We need to somehow have the idea there is self of life and outside. There's inside, outside. And this is in fact one way of drawing this here. In this way of drawing it, the image comes, and I should have put that on the first one here. This is the, oops. Sorry for the animation. What's going on? There's something stumbles over the animation. I'm sorry for that. So that's from David Gutzel, and I cannot show you. It's a simple cell. I cannot show you his name. I will hopefully get it back later. But something is wrong when I put that image on. And in this so this is called a membrane, and this is called a lipid bilayer because it's a particular molecule, lipids, fatty acid. It's a bilayer because the fats hang there from two ends. Essentially, this is something that is protecting you from the water getting into the cell. That's as simple as it is. So you have something, you cannot put plastic around it. So not because plastic was not invented by life. This is maybe also a reason, but the problem is, why can you not put something that is completely sealing off this cell?
Because it should be semi-permeable. Some things need to be.
And nutrients, yes. You need to get food in, right? Mm-hmm. For instance, if the self sits in this one here, you need to get somehow food in. But that means, and you need to actually also get some stuff out. So anything that you no longer need, let's put it like that, is put out. You need to secrete. And you need to communicate, most likely, with the environment. And communication with the environment helps you, for instance, to find sources where better food is, or to move around, or whatever you do. But clearly, this has to be a protection against the environment, but it should somehow be semi-transparent, or somehow transparent. So somehow things have to get in and out. Okay, and in and out, ideally, you use a material that is so you could imagine that you have like a door that you actively open and close. And we had that in the last semester. Proteins do exactly that. Proteins are these doors, so-called membrane proteins. They sit really like gates and gatekeepers. Both they open the gate and they are the gate. You can really see that, and they are the gate both for small molecules such as ions. And for larger constructs. So there are really gates that open up. But still you want to have a somehow semi-transparent membrane. And that is what you see here, surrounding a simple cell. And here's the Gutzel citation. So now that gets to the next point. How densely packed are cells? So I said that in me there are about 67 trillion cells, in every one of you as well. So we have about 60 trillion cells. How densely packed are they? How do you imagine a cell? Okay, let me put it differently. The image is you take, and now let me get back to plastic just for the analogy. I don't mean plastic. You take a plastic bag, you fill it with water, and you put some stuff in there that floats around. Is that your image of a cell? So I take a simple, ideally transparent plastic bag, because then I can look inside. So I have a transparent plastic bag. Again, I know the cell is not transparent, is not plastic. It's a membrane, a lipid membrane, but I cannot sort of create easily a lipid membrane. So I take a plastic bag, fill it with water, and then I put some stuff in that I call the nucleus, all the kind of things that swim around. Is that your image of a cell? Are you happy with that image of a cell? So some people have been with me for longer, and they know when I ask a question like that, the answer is always no. Is that why you smile? Yeah. It is true. So this is clearly the way. Before I studied molecular biology, so when I finished my study, I had not heard a lot of molecular biology. That was my image. But it's very, very wrong, because the cell is entirely densely packed. So again, drawing from David Gottsall, and he is just, so his art is to present realities of life that you cannot make a photo of in drawings. And he researches these drawings very long. So this is a real, I don't know whether you call that art form. This is an art, undoubtedly, but essentially he sits in a science institute. And really, he writes reviews. He really is absolutely a scientist and an artist at the same time. His drawings are absolutely reflecting what we believe is the reality. And that is, for instance, the drawing of the density of what is happening in a cell. It is true that everything inside of the cell is surrounded, all the proteins are surrounded by water. They are in droplets of water, but you can't really see them because it looks like a watch. Cogs and a wheel, so everything fits together, right? It's very densely packed, and the density of a cell, in fact, is like a solid state, like something, like a table or something like that. And that really is true. It's a high density, nevertheless, things move around there, and they are sort of swimming in water. Both are true at the same time. The way to imagine it, you need a very thin layer of water to make things move. That ultimately sort of explains this idea. Or this paradox, what may appear as a paradoxon. So again, DNA to RNA to proteins. So how many are there? So the smallest organism, Mycoplasma genitalium, sort of has 500 genes. So that means we have 500 different proteins, and that sort of make up the entire diversity of that simple organism. It's a bacterium. So we get here into other bacteria, and you see the line of bacteria goes to E. coli, bacterium in the gut, with sort of 4,000 genes. And that's where the world of so-called eukaryotes starts. Bacteria are creatures that essentially have only a membrane, and then stuff swim around. Eukaryotes, coming on from here, they have a substructure. They have a core. Inside of the core is the DNA. Okay? In the bacterium, the DNA swims around. In the eukaryotes, they sort of are in one particular compartment. And there are other compartments that have special reasons to be there, and I will get back to that in 2 weeks from now. Now, the first eukaryote here is yeast. That's Baker's yeast here. This is a unicellular eukaryote. Unicellular simply means they survive as single cells. So they are creatures just like bacteria. Most bacteria do not live in colonies, so they live on their own. And yeast is a unicellular organism that does live in colonies, but it can live completely alone. And here we get to the first organism, the fruit fly, that in fact is a eukaryote with many multicellular eukaryotes, so different types of cells, worm, a weed here, rice, and human. Now when you look at the number of proteins, you may stumble over the fact that in fact something so small as this tiny worm here has, in terms of number of proteins or number of genes, a similar number as human has. And now you may ask, why is that? How can I believe I'm more complex than this worm? I sure believe I'm more intelligent than this worm most of the time. And I sure believe that I'm sort of a more complex being. Why is that not reflected in the number of genes? And the answer is very simple and very clear. We have no idea. Before the human genome was sequenced, people believed that worm would have about 16,000. It was sequenced just before human, and they came up originally away with 15,000 or 16,000. And human at that point was believed to be 100,000. And then the universe was fine. And everybody believed this was okay. And then just a few months before the final publication of the human genome, there was this press announcement, we made a tiny mistake. It's not 100,000, it's 30,000. So for biology, this is what biologists call a small mistake, right? That tells you, you may laugh about it, may call them dumb, but it tells you about the amount of novelty that you can discover within a few months in biology. There's so much not known. There's so much to know. We're so far away from saturation. So that was 2000, and then people believed 30,000. By now we believe it's more around 20,000 proteins. So again, we don't know how with these numbers we can reach a higher complexity. So again, there's something here, the RNA in between, but let's not get into that. Let's get into function. So function, those what I show you here are images of proteins doing certain things. And that brings us back to the first question, what is a protein? And in fact, if you now expect that I'm going to, in the next slide, give you the definition a protein is, this is not math, this is not physics. In biology, you're not getting any of that. In fact, so I've been working on proteins for 25 years, I cannot give you a good definition. Let's begin with the first thing. A protein is made up of amino acids, and amino acids, there are 20 different amino acids, so you can represent them as letters. Alphabet with 20 letters, okay? So proteins are stringed up letters. Whatever you call it. You can call it one word, one protein is one word, you can call it a sentence. Name it as you wish. The idea of a sentence makes sense because there's a substructure, there are regions in proteins, so whatever you say is just an aggregation of amino acids. So that's one simple definition. Another thing that we know is the moment we take this string of amino acids and put it into solvent, essentially water, it adopts a unique three-dimensional structure on its own. Most proteins float in water completely on their own. That means there's nothing else they need to know. They don't need help from anybody else. It's just the physics, the biophysics of the floating process. And that ultimately means that the information content for how they look in 3D structure is in the sequence. Okay? Or in the interaction, at least, between the sequence and the environment, which is water. And in fact, it's also true, and that was originally shown here by Christian Anfinsen, who got a Nobel Prize for that. It's also true that, in fact, function is determined by the sequence. Ultimately, function is determined by structures. Function is determined by the shape. How the protein interacts is determined by the shape. Here are just a few, that's a slide from Christina Rengo, that is now over 20 years old. So it's the first time that Christine and Janet Thornton here put together a view of the diversity of the space of different protein structures. This is just one way of looking at it. The green is always, so there are two, for those of you who did not participate in last semester, so there's secondary structure, there are two main secondary structure elements, and simply when you look at a protein structure, you can recognize repeated patterns, and there are two types of patterns. One is called a beta strand, that's shown in green. In the beta strand, when this is your protein that sort of folds up, this protein is too short. This doesn't get longer. So when they fold up like this, so beta strands are essentially things where two stretches come together here and they bind. Okay, and then maybe there's a third one coming, and then they form a three-mer stretch. So three bind, and so it's these two and these two here that bind with one another. That's called a beta sheet or strands, and that's shown in red. In green, the red things is the second main secondary structure element. It forms helices, just like this one here. Well, anyway, you get the idea. Okay. Like the DNA double helix, but in this particular case, it's a single helix. An example you see here, showing, and here these two helices are binding. In fact, 1, 2, 3 is three helices binding. Up there you have another two helices. Here you have sort of helices all around it. Here you have the beta strands. This is a propeller fold. It looks like a propeller. The horseshoe because of obvious reasons. And you can already see from these words, propeller, horseshoe. So the way they are described is you look at them and they look like something that reminds you of something that you have seen before. And you see a lot of these simple machines in the world of proteins. Here are some of these membrane proteins. Those are channels, so that let ions through. This is a particular protein here that is related to arthritis. This is another protein, it's a glycoprotein 120 that is related to AIDS. That's the virus shell protein. So proteins essentially come on different sizes. They adopt a certain 3D structure and they start from a sequence. Now, that gets us to the next issue. We could argue a protein is something I go to the genome, I take out from the genome something I translate it to a protein, and that is a gene, that is a protein. And that is not quite true. Here's an example of a protein where this breaks down completely. It's the so-called ATPase. It's another one of these giant membrane proteins that in fact creates energy. But in fact it does all kinds of other things. One of the things it does, it has, and this is not easy to see here, but it has a very long, so two helices that are wound around each other like this, and when these two helices turn, then they can actually drill a hole. And this thing is just doing something like that. Okay, it drills a hole into membranes, it drills a hole into structures. There's a lot of substructures. So here are different. So it's a dimer, so it has to form from two different segments, and they stack like A, B, A, B, A, B. So they all come together from different subunits. The remarkable reality here is that the unit, the ATP unit as such, this protein that we think about as a protein, comes from two completely different regions in the genome. It's one protein, we think about it as one protein, it's one functional unit. It's expressed together at the same time as one thing, but it comes from very different places in the genome. So that breaks down this idea that proteins are things that are lining along the genome. So what a protein really is a little bit more complicated. But essentially, thinking about it as a linear sequence is certainly for this course good enough for you in that sense. Now, what you typically do if you begin with a sequence, and again that was in last semester, we have one sequence of a protein here, you don't know what that is. There's another sequence in the database of which you know it's some particular enzyme called P450. So some have names that are sort of describing something, and some are names that are completely indescriptive. And this comes from a species spearmint. This comes from a species plant peppermint. And now you wonder, what is this one doing? And when you compare the sequences, essentially you ask, I compare these letters one by one, then I can essentially say that's what is called an alignment. I find the optimal superposition of these two strings. I find the way in which I get the highest number of identical letters. So I just slide them by each other, and there's an algorithm for that, dynamic programming. There are hacks around hashing, and again that we did in last semester. But ultimately you can compare them, and when you compare them you can find out, oops, in fact, that this one here is the cytochrome P450 in the other plant. Okay, so by knowing this one experimentally, you can infer that this one does the same thing because you looked at this one-dimensional string. Okay, this ultimately is the way you use these sequences, and the sequences are meaningful. Just a few facts: how many proteins are in human? About 20K. I already said that. The length is, well, the shortest are around 35 residues. This is essentially 35 letters, and the longest one is 30,000 residues. So there's an immense spread. The median is around 400, but when you sort of look at the distribution of proteins, you see that putting a median into a distribution that sort of essentially occupies the entire space between something like 100 here and 500 or 600, just the word median is misleading in this sense, right? And you see that there's a very long tail. In fact, you don't see it because this is just like the internet. So the ends are, there are still things over there. They add to very little. There's this big data issue in terms of the growth of the databases. So when our group moved from New York to Germany here, there in all databases there were 8 million proteins known. 4 years later, when I did this slide, there were 35. So this is double every 2 years, right? and everybody says, okay, this is like computers. So we double every 2 years is a big number. I always thought that computers grow the fastest, but it's not shocking me. Okay, computer growth slowed a little bit down. We're not quite double every 2 years, but it's not that far from the kind of things you guys are used to, and that's true. But then remember, we have to sort of compare, right? So for everyone in the database, we have to find all the ones that are similar to it. So that actually means we're not going to look at the 8 million. We're going to do comparisons. We're going to compare everyone against all the others. So that means in 2009 here, we had 6 by the 10 by the 12. 4 years later, we have 1.2 by the 15. And now, actually, for the amount of operations we have to do, for the processing, there's a 20-fold increase. And that, in fact, is a major problem for computational biology. So the biological data grows much faster than the computers. So every year, when I started in this field in the 80s, every year the computers got faster and we could do more next year. And now that has reversed completely. So every year we try a new hack. We try to sort of speed up, we all kinds of things. But we're really fighting against that, we as in the field. Nobody has a solution for that. This is very, very simple numbers, right? The only hack around is you avoid some comparison. You reduce the number of things to compare because you filter out things that you believe are not necessary. Here's another sort of side aspect of that. In terms of what we needed here, when we came from New York, it was 5 terabyte. I actually transported them carry-on in the plane. In disks, because I just, that was important, the entire lab stuff. 5 tera, those were, in those days, were 3 disks. Even today, so this is 5 years ago, right? But even today, 150 terabyte, you do not take as a carry-on. Things have, there is the sort of same issue here on the level of the amount of data. And by the way, so when the human genome was sequenced, that took about, this is difficult to gauge really, but say 5 to 10 years to determine the human genome. Today you buy a machine that looks like a printer, same size, it comes in the same package, costs a little bit more money than your average printer, and essentially it does a genome in a day. This thing cranks out, if you take the raw data, this thing cranks out every day 8 terabyte. Okay? So which again is today something, you all know that. You want to feed this thing with 8 terabyte disks all the time, right? Okay, today you can, tomorrow you can, but at some point you're going to actually see, 365 days later, you're going to see a lot of 8 terabyte disks in front of you and actually don't know what to do with it. So it creates all kinds of problems. How do you believe these large sequencing centers in the world exchange data? Given all what you heard from me now. So I believe, again, the people who haven't been with me, it's very clear that the way I ask now, you will not say internet. That is not the answer we're looking for. Doesn't work. In fact, there is an example where a node from the EMBL in Australia began to download some part of a relatively small database, and 3 months later they switched it off. And that's a tiny database. It's not these sizes. So the way they do it is, of course, FedEx. So they really FedEx around hard disks. That's the modern way, 2018, in which the top-of-the-line genome sequencing centers exchange information between each other, between Shenzhen and Harvard or something like that. Okay. Proteins consist of units, so-called domains. Here you see an example for that. So this is a fraction of this. So you see parts in other parts. This is a protein with 2 domains. Most proteins have, in fact, more than 1 unit. We know that the same sequence essentially always, so again there are tiny issues here, but essentially the same sequence always falls into the same 3D structure. Does the same human always have the same sequence? Essentially it does. And the same sequence, again, we are all back to the amino acids. Here are the 20 amino acids. They look chemically different. Some have positive charges, some have negative charges, some are polar, some are hydrophobic. Hydrophobic means you want to be away from water. And some are hydrophilic, so you want to be in water. What is a protein structure? A protein structure is a shape. Here is a protein structure of two proteins that are interwoven. I've shown this slide, or this and the next slides last semester. And ultimately the point here is I'm going to show you a bunch of different images of protein structures, and they do not all look the same. So what is a protein structure? I will get back to that answer. One is this image. One answer, okay? Here's another image of the same thing. Now the rods represent the helices that I talked about before. These in this particular protein here, they're long helices. What you see on this image that you don't see on that image here is immediately that you have some symmetry. You see that the red, I mean most of you, even if you haven't seen a protein structure before, you will recognize immediately that red and blue look somehow alike. It looks like something that is that you can turn into the other, right? Like somehow mirror image of each other. In fact, they are. But you have to be, some people can see that from that image too, but you have to really be much more into protein structures than most of you most likely will be. So in this representation, it's a different representation. It's the same information represented in a slightly different way. And this representation helps you to see something. Now, ultimately, what is a protein structure? It is coordinates in a 3D structure. Okay? The way I visualize it has to depends on what aspect of the protein structure am I focusing on. Here, I'm focusing, for instance, on major secondary structure elements that are maintained or stabilized by some bonds. And I visualize them as rod. Okay, and I say there is a set of amino acids that have some commonality. They're part of a helix. Okay, and that is what this image shows. And that showing that, reading that in this image, helps me to understand something about that structure. For instance, that they sort of move actually as rods when they function. Now there is another aspect, and that is already seen here. Since these two come together, you already see that one aspect of protein structure is not only how the red or the blue or the red look, right? It's also the fact that they come together. It's also part of the structure. In fact, this particular protein here, you will not see act the potassium channel. You will not see act as a monomer or as only the blue one. Okay? In the next example, I show you a molecule, so-called ADH, alcohol dehydrogenase. Alcohol is clear to most people in the room. Dehydrogenase essentially is what you need to do, what your body needs to do if you put this alcohol inside of you and you don't want to feel bad. So you want to sort of digest the alcohol. You want an enzyme that essentially eats up the alcohol. And that is essentially alcohol dehydrogenase, ADH in short. In particular, the particular bunch of these enzymes, this particular type happens to be called ADH5 and comes from human. Now, ADH5 here shown as a homodimer, that simply is that protein. So the sequence I showed you before in 3-dimensional structure. But functional, it is only if it forms a dimer. So if two of these come together, and that's shown here. So these two.
I have one basic question. I mean, what makes two human different in terms of protein? Is it the concentration of each of the proteins we have?
So what makes me different from a mouse? Is that what you're asking?
No, two humans different in terms of protein. I mean,
two humans. Yeah. So in this particular case, in fact, that is why I put this protein up. What makes us differ from one another is not only the gender. But most likely I have this ADH5 and you don't. So this is one of those proteins where in fact subpopulations differ.
when we say that we have this number of protein, I mean number of proteins in a human, so that shouldn't.
So there are some examples. So there are some examples, so some famous examples of enzymes that some subspecies have. For instance, most northern Caucasians, white Caucasians, essentially have lactose. Have the enzyme that have a version of the lactose that allows are expressing an enzyme that so when we are babies, we all enjoy milk, and then for some of us, for most creatures, for most mammals are the ones that survive on milk, but for most mammals, after the baby time is over, when they're grown up, that enzyme is no longer expressed, meaning that milk becomes poisonous to them. What you know as lactose intolerant. That is, in fact, for almost every mammal on this planet, the natural reaction. There is an exception. Caucasian white people, such as I, we express the same protein when we are adults. I can drink milk and still feel okay. For many people, that's not the case. So sometimes it is really true that between two different types of populations, a particular protein is not existing. There are very few examples of those. This is one, and in this particular case, most likely you actually don't have the gene. It's not only that you don't express that protein. For the lactase, it is that we both have it, but I express it as adult, you don't.
So when we say that, so we don't have the same number of proteins we can...
No, wait. So I said these are exceptions. I also said, I gave you some slides that said human have 20,000 proteins. I gave some where I said human has 21 or something like that. So this number is not, we cannot go into you and I and count the number of proteins. This is a wild guess. What we do know now is that there are cases of single entire proteins different between two individuals. Okay? That is a very big surprise, except for these exceptional stories we have been knowing about since many years. But that in fact really any pair in this room could differ by one entire protein other than sort of things like ADH, that is actually new. That is surprising, and we still don't quite know how relevant that is, because we still cannot count the proteins. Typically, the way, and I will get back into that toward the end of the lecture, where I will in fact have a lecture exactly on the question, the differences between us, between the proteins that we have. What do they affect in terms of function? I will talk about that. And ultimately, the issue, the way we see that, or we imagine that, is that most of the differences between you and I, again, other than between a man and a man, or a woman and a woman, is that on every single protein that we have, so essentially we assume that both of us have 20,000. So here's one you don't have, but then you will have something that I don't have, that I don't have, okay? But essentially we have 20,000, and the difference is that you have a slightly different amino acid in each one of these 20,000. From the two of us, from the two of you, and from the two of you. With the exception of that, since there's some chromosome difference between men and women. That's another exception. But other than that, ultimately all we believe is that in the sequence here, the sequence I haven't written, but in the sequence that I put up, between the two of us, in every one of these sequences, one letter is different. And will that have an effect or not? And that is exactly what we currently cannot answer experimentally, only with the kind of methods that I will present in this lecture. And one of my final lectures will exactly be sort of the wrap-up talk, getting into that answer. Any other question on that issue? Back again to that point. The point here is they need to come together in order to do the enzymatic reaction. Okay, so here's an enzymatic reaction. It sort of eats some alcohol. But more formally, what is protein function? So in this particular case of the ADH5, some of that is already written, recognizable by experts in the name. So there's something about the substrate, alcohol. The hydrogenase means it cuts something. then there is sort of a little bit, there are different names here used in different contexts that bring class 3, so you have a little bit more detail in this particular case. Here, you in fact have more, again, is another name of the same thing, where you bring a little bit more the process that is sort of setting this off. By the way, here's the sequence. And now you can call all of this sort of function. But then there's more to the story. And here's this is from Protopedia, so that's a Wikipedia-like attempt to classify proteins. And you see in this description, so you can read it, there's a lot of information. The important part that I want to bring home here is this lot of information is not such that you see one single number. Here is one way for enzymes, and we will get back to that. There's a classification that is enzyme specific. So enzyme is a particular type of protein that cuts or mends certain bonds. In fact, it's a catalyst, so it allows reactions to happen at a rate that is much faster than they would normally react. So one way of thinking about that is, say there is a certain reaction that would take longer than the time the universe had so far, right? And with an enzyme, the same reaction would happen in a second, okay? So speeding up the entire history of the universe into a second. A reaction that would happen, most reactions happen. Remember, in chemistry there's always some equilibrium. So if you give things enough time, at least the reaction is going to happen once in a while, right? And the enzymes just enables it. Another way of thinking about it, it pushes reactions over an energetic barrier. So it allows you to sort of jump over a hill. You know, some of you may know it from cars, catalyzators in cars. But enzymes essentially are things that allow processes to happen and make processes much faster, and they increasingly are used. So increasingly enzymes from bacteria, from the bio world, is used for large-scale processes to produce all kinds of things, such as drugs and other things. Now, all the enzymes that are known are classified in a schema called the EC number, where the first digit in this EC number sort of is the large scale, the overall reductase, so does it cut or mend a bond, essentially is what this number says. And there's 6 of these classes. And then we get to more detail here. The more numbers you add, and we get at the end here to the aldehyde dehydrogenase, and the numbers in between are essentially the type of bond, the type of acceptor, so more details are added, right? Okay, so this is one aspect of what the ADH does. Its function, it is its characterization, and it is the attempt to classify all enzymes into one number. But it's not the entire thing it does, because actually, here is written, it's the 4th step in the metabolism of fructose. And breakdown is what metabolism means, right? Fructose is just one type of sugar. It's a more healthy type of sugar than the white sugar from fruits. And so what this means here is this is cutting a bond. Okay, that's part of the function of a protein. But it is part of a chain of things that happen. Fructose is coming into the body, is broken down. It's not one enzyme that is going to do that. There's a bunch of things that are needed, and things, there's always proteins in this world. So there are several proteins needed to turn whatever you eat into whatever your body needs, right? It's not one enzyme. It's a process. And in that sense, its function is to be part of this process. It breaks down fructose, okay? It catalyzes a reaction to glycerol, which again is part of this part. That's the chemical formula that describes it. Anyway, but the point that I wanted to make here is, in order to describe what it does, part of the role it has is being part of a long pathway of things, and every blue one here is a protein in this entire pathway of fructose. And you immediately see there's a lot of blue. and there's an organization. So it's not a straight path. One does this and one does that, right? There are circles in here. There are all kinds. It comes from KEGG, a database of these pathways. Now, there is another attempt to classify proteins into an ontology, clearly, obviously called the gene ontology. And the gene ontology essentially has three different, and I will get back to that, three different components. So on the one end, it classifies proteins by their molecular function. That is the chemical reaction that I showed the formula for. One is by the processes, saying, well, there are all these processes and this is part of that process. It's a different ontology. So we have three different ontologies. One is the molecular function, one the chemistry, one is the processes, and the third one is essentially where the cellular component. So where in the cell are you? Where are you doing that? And that, of course, relates to the when. Now, one way of defining, so ultimately what it means is what you consider to be protein function essentially depends on what you are studying in your research. Are you sort of more on the chemistry side and really want to know how the atom is bound, want to know that this is an enzyme, or when in the cellular process it happens, when in the development of an organism is this protein expressed, is it related to disease, is it a dominant or recessive, is it sort of genetic, and all of these are different aspects of function too. So I said process and molecular, but there's much more. So the function of a protein is not easily defined, because all of this is part of what the protein does. Okay? And it depends on what you study, what you consider the most important aspect. There is no one most important aspect. While for structure, really, there is an atom in space. There's no second guessing of that, right? In that sense, function is much more complex. That's not even a definition. This is sort of one way of defining it. Anything that happens to or through a protein is function. So the repetition of that definition here. And it may seem that I'm not getting through my slides today. Now, our special angle in this is sort of the combination of evolutionary information with machine learning. Evolutionary information that you get from families of proteins, related proteins. Now, one question that we can ask, for how many proteins, so one thing that we see here, databases explode, and that's a very old slide, just to say that databases have been exploding for more than 20 years. So that's the genes here, that's proteins with experimental annotations, and that's proteins with known structure. So where we have experimental information is growing much slower than where we don't. And that has gone much worse ever since. Ultimately, when we ask how many proteins in human do we know the function for, then the answer is, and that's one answer, 10 to 50%. And from this precise number, you already see that there is a lot of lack of clarity. Ultimately, whether I say 10 or 50 is whether essentially you consider this one here to be essentially red, or whether you consider that one essentially to be red. If you consider that essentially to be red, then essentially we have 50%. It has to do with something such as precision of what we mean by function. Is it enough if I say that this protein actually has something to do with breakdown of alcohol? Or do I really want to know exactly how it does that? In which process, right? If I call this one here an aeroplane, then we know 50%. If I believe that the detail I need is to call an aeroplane is this level of detail, right? Then we know 10%. This is why there is a range here. So it is in fact unclear. We cannot clearly say how much we know. But clearly, for most proteins, we have no idea what they do. For most human proteins. Now, the evolutionary story here. I said that Epstein showed that from protein sequence you get three-dimensional structure and function. In some sense, when you think about it as from the perspective of physics, the types of problems that physicists typically look at are these type of problems. So at some time point you have the ball and you want to predict where it rolls, right? And it's fairly easy. It's even easy if you make a tiny mistake in the initial conditions. The ball will still roll at the same point. While if you have this type of system, you immediately see that tiny problems here in the initial conditions, and you no longer know in which direction it's going to move, right? And the protein function is a problem of that type. Because a point mutation, and that brings us back to what you asked, a protein between us, there's one difference in the amino acid. And now the next question would be, does that actually change the function? And it can. So we have many diseases. In fact, at this point, we know about 2,000 diseases in human that are caused by a single letter change. That single letter change, whoever has that, gets sick. 2,000 cases of those we know. We know more than 2,000 people, obviously, but this is just one example. They are exceptional. Very exceptional cases, but there are 2,000 different diseases of this exceptional type. Whether substrate is bound or not may change function. So is something already around it? An environmental change, is DNA close to it or not, will change function. So clearly the prediction of function is that type of problem, or the prediction of structure, where ultimately if we think about it, we need to know the history of the entire molecule. And if we need to know the history of the molecule, this is a very difficult prediction task. But, and that's the point where evolutionary information comes in. What I show here on this axis, number of residues aligned, percentage sequence identity. And it's an alignment between two proteins. So I compared protein sequences and I asked how many of their letters are identical. Okay? And I look, for instance, at all the cases where I have 100 aligned between two, and at least 33 are identical. So more than 33 are in the blue. Okay? That means that, turn that around, you can change 67. So 100 minus 33 is 67, right? You can change 67 and you still get the same structure. Because every pair in the blue essentially has the same structure. So now comes the surprising reality, and this is a question that is only for the people who do not know the answer, which I hope is highly sampled by the people who have not been in last semester's lecture. So to you the question is, here's the next observation. My observation is I go to the lab, I say 67 and 100 I can change. I go to the lab and I change 5. Just randomly, okay? And I will change structure. How can that be true? That I can not even change 5 without affecting structure? And nature can exchange 67. Any idea? So if your answer would be that I'm an idiot, fair enough, I take that blame. I'm really not gifted. That's why I became a theoretical physicist at some point in my life, because I was not good at experiment. But so that's part of the reason. But there's another answer. How come?
What is the question that?
If nature can change 67 residues, I can only change 5. Again, I'm an idiot, but nature's more intelligent than I. That's true. But there's something else. How come that we see in the record 67 residues can be changed between pairs of proteins, and they do when I observe this pair, they have a similar structure?
Maybe because we can't create the environment that nature provides.
That's an interesting point, but the environment essentially is water. So all I need to do is throw them into water.
But you can't, create the actual environment in your lab.
Yeah, you are sort of escaping with what I would, as a scientist, call the religious argument, because what you're saying is ultimately there's something that makes it different, and that is true. But put your hand more on what this something is. Be a little bit sharper. I'm not saying that you're not sharp, but don't get me wrong. I'm not in any way trying to attack you. But it's too. Yes, you're in the right direction. Pinch or get closer. Zoom in. Yeah.
I think your probability is really low because, for example, if you have a 150 long protein and each amino acid could have like 1 of 20 amino acids, then you have 1 in 20 to the power of 150, which is really low. So
Yes.
you need to change 5, but you need a lot of time to figure out what 5 it is on.
That's exactly the point. The probability of what you call a neutral change. So neutral changes, I say these 67 that don't change structure, I call them neutral with respect to the structure. And the probability of having neutral change is very low. How come we observe them? Because nature had enough time. Because, in fact, there's a selection mechanism, and that is sort of the system that you talk about. What I don't have in the lab is the selection system. So one of the Nobel Prizes this year was for an evolutionary selection system. And that is, in fact, the selection system in the lab that she proposed. Francis Arnold. There we would sort of recreate that environment in the lab, but the essential point here is that you have a selection system that if the protein does not have the same 3D structure, if it does not function the same way, then the organism will have a problem and the organism will not be selected. So that change will not survive simply because it doesn't work. Okay? So all that you see are the ones that did work. That means they are, if they were originating from a change, they were neutral. Very unlikely, but that's the only thing that survives. And that's simply the selection mechanism that you see. And many million years of selection, or in fact a billion years of selection, allows you to really sample a lot. That ultimately is exactly the answer. What we see here are two things. We see a system that provides for free a distinction between what is a neutral and not neutral. Well, we don't have the non-neutral ones, but at least it's an identification of the neutral ones, okay? And we see that something survived. Now what we, for instance, see, and here is one particular family, as so-called SH3 is an important enzyme for signaling. Signaling means essentially it begins a cascade of events, kinases. So it sits on a receptor at the outside and says something is going to happen. And this signal is transferred into the nucleus and some new protein is made. This is what kinases, for instance, trigger. That's a large family, is a very old slide. The only important point is that there are essentially 4 columns here. This one, this, and this, that are essentially conserved, meaning that the entire family has sort of the same type of residue at that particular position. When you look at this column, on the 3D structure is here, this one is here, this one is here, and the other one there. So these are 2 different sides of the same molecule. And the important thing that I wanted to point out here is this one and the first one here, they come close in space. Okay, now nature acts upon this as a 3-dimensional object, not the sequence, right? What nature sees is the folded protein. Constraints, anything that goes right or wrong in evolution always happens on this 3-dimensional object. This 3-dimensional object, anything that happens to this residue will have an impact here. Whatever happens here will have an impact there because they are close in space. Okay, if this suddenly begins to be too big, then it pushes the other one inside or something like that, right? So the shape will change, or may change, of the biophysical features of the. So say this is a positive charge and this is a negative charge. If I switch one of those, there would be repulsion suddenly, right? So you would immediately see that something that could happen here could have an effect here. Now this in turn means that if I take out 5 consecutive residues from this part here, so sequence consecutive now, in the sequence neighborhood of 5, and I look at this profile, in that profile I see something that is causative from here, which in sequence is very far away. Meaning, from this profile, from taking out these 5, these evolutionary profiles, so all of these here from the 5, I see something that may have happened here. I mean, I'm not seeing it directly, right? But I see evidence on it. Something that happens far away in sequence here, not far away in 3D, but far away in the linear sequence. This to here is a distance. And from cutting out what happens here, that what happens here is constrained by what happens here. So I see effects that are from far away in terms of sequence space because the profile that I have, this evolutionary record, has acted upon a 3-dimensional object. Okay? And this essentially is allowing me, you can imagine, to sort of transfer this type of problem, the chaos type of problem, the non-solvable kind of problem, by averaging over different species, over the evolutionary record of different species, over what is a neutral, unlikely but neutral variant, and I can use that to sort of turn this into that type of problem and solve it, and for instance, predict secondary structure by using this kind of profile as input to secondary structure predictions into a neural network, into machine learning devices. And again, I'm going to show you several of these over the course of this semester. If we succeeded to do that, we could have a variety of advantages of medical applications, molecular biology, and many others. And this one here is, in fact, these kind of things are increasing every day. I'm beginning to run out of time, but it's a slide from Ana Luisa Arruda that just shows for one particular protein structure, one particular protein, the leptines, we believe that has one particular function, how this particular function can in fact involve many different cell types, regulate appetite, control metabolism and energy homeostasis. Here it activates the immune system, here it does something with glucose, so it does some very different things. Because it's in fact, it does the same molecular biology, but it's involved in different pathways again. So the same protein in different cells can do different things depending on how it is regulated. Function, and I believe this is where I'm sort of going to begin to fast forward. A lot of function is motion, molecular dynamics, so there's motion on different time scales. Here is some, so this is a regular protein that binds a substrate, there's substrate coming in here. And you see that this entire protein is not, so we make these images of proteins as crystals, but you see that actually there's a lot of movement, and you see in particular, so the more you get to the center of this, the less movement, but on the outskirts, on the outsides, you have sort of the more floppy regions, and then you see that the binding of the substrate here, of this ligand, is in fact decided by sort of reaching around and by trying different conformations. Meaning that really the protein itself adjusts to the binding, the molecule that it wants to bind. And passes it around. The movie that you see here is the source. The movie that you see is done by molecular dynamic simulation. So once you have the 3D structure experimentally, you can put it into the computer, and then you can really simulate the motion. And in fact, you can simulate this only in the computer. You cannot see this in the microscope from happening. Here are some faster motions. This is essentially pushing these ions in or out, or avoiding that these ions that in fact reach inside here. That's a membrane protein, and ion by ion is sort of lining up and waiting to get inside the cell. And when this thing here opens up, one ion will be pushed further. And you see again in the shape of this thing that it sort of holds exactly 3 ions there. And if the ion would have the wrong charge or the wrong thing, it would never get there. So, and again, in the context of the entire simulation, this is from Punta, Marco De Vivo. So this is relevant for drug design. And here's another example.
When it finds its binding site
from David Shaw's.
Center of the water, it's fitting very neatly in there, which is what the drug really wants to do, among other things. So here's another one. This is also binding to SAR kinase. It's a drug called dasatinib, and it is an actual drug that's approved for leukemia. And it's known again where that binds, and this is another experiment being did where it is thrown into the box. It is harder than that, but put it in the box, and it fairly quickly, in this case, found the rough right area. It's going to pause here, and it's actually kind of interesting because, well, maybe it already did it. A little groove opened up here, and it got distracted by it for a second, but then eventually it goes to exactly where it's supposed to. And then this is a different kinase. It's the epidermal growth factor receptor, which is it's actually a family of 4 different proteins that are implicated in several different indications, but in this case, for this drug, the drug is approved for the treatment of breast cancer. It's called dasatinib, and I guess it's hidden right now. This is the last one I'll show, but the drug's going to search around again. and it's going to find the right place. Again, there are a few false places that it is before it finds the right one. One very interesting thing about this is, this isn't like a new finding, but it's what's called induced fit. In the process of having the drug molecule fit into a protein target, it's not just that the drug itself moves around, it's also that the protein moves around, making space for it. And the induced fit part is when the drug actually gets there, you can actually change the shape of the protein locally. And many methods to computationally identify the drug binding site and look at how well it actually works as a drug assume a rigid protein. They might actually start with several different snapshots of the way the protein could be, and then what's called dock all of these little drug molecules into it, see where they could fit, not through the actual dynamics, but just kind of moving around like this in all the possible rotations. But there's some kinds of drug binding which probably occur in areas that aren't even pockets. They're not, usually these kind of drugs bind to little depressions, and they fit very nicely, and they have certain kinds of chemical interactions. But in some cases, we don't think they're there. This has been postulated. We're seeing some examples that I haven't shown you where it appears to create its own pocket where you couldn't have predicted where it was going to bind and then bind in there. But this is all very, very preliminary, intriguing from the viewpoint of drug design, but early stage. So switching to...
Okay. And I believe I also have to switch gears. So David Shaw, in fact, Anton, you saw the name Anton up there. This is a special purpose-built machine. At this point, there are several generations of Anton. And they are worldwide by far most advanced in doing these molecular dynamic simulation. In fact, they have, within a few years, with very focused funding from David Shaw, really beat what the public expertise has done over decades. And they have gone very far. And what we can today do, and what this kind of thing can do, is what you saw. It can simulate how the drug binds in some cases, but you always need to start from a 3D structure. Now, I will briefly go through. There's no point. There's no point in going through 40 slides in 2 minutes. I'm going to cut it here and see what of these slides I really deem absolutely important. Let me just make this one point and go through the next slide. So I said that a protein adopts a unique 3-dimensional structure and that the shape, the structure, is relevant for function. And that's sort of what this image, order means function. Now here's an image where we talk about what is called in the protein world often referred to as disorder. So we have a stretch here that sort of flops around. Flops around means you look at it at different time points and you do see different things. Okay? Then it binds to some molecule, and upon binding you induce a regular structure. So here it looks like the helix. Now if you if you could look at it with a microscope, again you can't, but if you could, you would always see the same. So upon binding, suddenly a regular structure is induced, and it seems that in eukaryotes we have a lot of this type of disorder, and that in fact is related to function.